Hello students, welcome to biology class. This video is in continuity with the previous video of chapter Transport in Plants. The objectives that we are covering in this video are osmosis, plasmolysis, osmotic pressure and osmotic potential, TURGOR pressure and wall pressure, imbibition and wilting. Starting with the topic osmosis. Osmosis is defined as the movement of solvent particles from the region of high concentration to region of low concentration. Osmosis is a passive process and occurs without any expenditure of energy. It involves the movement of molecules from a region of high concentration to lower concentration until the concentrations become equal on either side of the membrane. There are two types of osmosis. First one, endosmosis, and second one, exosmosis. Endosmosis refers to the inward movement of water through the semi-permeable membrane when a plant cell is placed in a hypotonic solution. This is known as endosmosis. And exosmosis, when a plant cell is placed in a hypertonic solution, the solvent molecules move outside the cell and the cell becomes flaccid or undergoes plasmolysis. This is known as exosmosis. Now discussing changes in a plant cell when kept in solution of different concentration. There are three kinds of solutions on the basis of relative concentration of water and solutes with respect to their concentration in the cell. These are isotonic solution, hypotonic solution and hypertonic solution. First one is isotonic solution. The solution has the same concentration of water and solute as is inside a cell, or the external solution balances the osmotic pressure of the cytoplasm of the cell. The cell shows no change in this. Hypotonic solution. A solution has a lower solute concentration than inside the cell, or the external solution is more diluted than the cytoplasm. When a plant cell is kept in a hypotonic solution, the water moves in and the cell becomes turgid. Next is hypertonic solution. A solution has higher solute concentration than inside the cell, or the external solution is more concentrated than the cytoplasm. When the cell is placed in a hypertonic solution, the water will leave a cell by osmosis and the protoplasm will shrink and the cell membrane will be pulled away and shrink away from the cell wall. In this, the cell becomes flaccid and is said to be plasmolysed. Next is reverse osmosis. It is the expulsion of pure water from a solution through a semi-permeable membrane under the influence of pressure higher than the osmotic pressure of the solution. It is used in removing salts from saline water as well as extra purification of water. Reverse osmosis is the process which is used to remove ions, mineral, chemicals and other impurities from drinking water. In this process, greater pressure is applied, forcing the water to travel through a semi-permeable membrane in opposite to natural osmosis. Reverse osmosis works on the same principle as osmosis but in the reverse direction. In this process, direction of water flow is reversed by applying greater pressure. Reverse osmosis involves the application of pressure on one side of the solution, where a semi-permeable membrane is placed in between the solutions. This membrane is used to filter out contaminants. Due to the presence of membrane, large molecules of solute are not able to cross through it, and they remain on the pressurized side. The pure solvent, on the other hand, is allowed to pass through the membrane. When this happens, the molecules of solute start becoming concentrated on one side, while the other side of the membrane becomes diluted. This is reverse osmosis. Next is osmotic pressure and osmotic potential. Osmotic pressure may be defined as the pressure required to prevent the net movement of pure water into a solution so as to prevent the volume of solution from increasing. The modern plant physiologists have substituted the term osmotic pressure by osmotic potential. However, it is to be noted here that osmotic potential is numerically equal to the osmotic pressure but is negative in sign, and it is denoted by psi. Thus, osmotic pressure is equal to negative osmotic potential, or we can say osmotic potential is equal to negative osmotic pressure. It is a colligative property of solute and is dependent upon the number of solute particles and not upon the nature of solute. It is to be remembered that the value of the osmotic potential of a pure solvent is 0. Addition of more solute makes the osmotic potential increasingly more negative, whereas the dilution of a solution with the solvent decreases the value of osmotic potential. Next is turgor pressure. If a living plant cell or tissue is placed in hypotonic solution, water enters into the cell sap by endosmosis. As a result of the entry of water into the cell sap, a pressure is developed which presses the protoplasm against the cell and the cell becomes turgid. This pressure is called turgor pressure. It is also called hydrostatic pressure during endosmosis. The turgor pressure increases gradually, and when it has reached its maximum, it is equal to osmotic pressure. Importance of turgor pressure. Turgor pressure is essential for plants to live and grow properly. Turgor pressure also aids in cell enlargement. Turgor pressure keeps the cell and their organelles stretched. Turgor pressure keeps the leaves fully expanded and properly oriented to light, helps in opening and closing of stomata. Turgor pressure keeps a check on the excessive entry of water into cells. Turgor pressure also helps in autochory of some fruits. Autochory is defined as the dispersal of seeds by explosive mechanism. Loss of turgidity leads to wilting of leaves and drooping of shoots. Next is wall pressure. The wall pressure is the inward pressure developed by the cell wall to resist or to counteract the turgor pressure. As such, the wall pressure is equal but opposite in magnitude to turgor pressure, except when a cell becomes flaccid. Next is plasmolysis. Exosmosis leads to release of water from the cell, resulting in the contraction of protoplasm. The shrinkage of protoplasm of a cell from its cell wall under the influence of a hypertonic solution is called plasmolysis. The process of plasmolysis is usually reversible. If a plasmolyzed cell is placed in pure water or a hypertonic solution, endosmosis starts and the cell attains its original shape and size. This phenomenon is called deplasmolysis. In this image, we can see when a cell is placed in a hypertonic solution, there is shrinkage of the protoplasm of a cell from its cell wall. Now the cell becomes flaccid. and this cell is said to be plasmolyzed. The next is imbibition. Imbibition is the phenomenon of soaking up of water by certain materials, particularly in dry or semi-dry conditions. Actually, it is the process of adsorption of water by the hydrophilic colloids present in the concerned materials. However, the cell wall and the protoplasm are also able to absorb water by imbibition. This phenomenon requires the following two conditions. First one, affinity between imbibant and the substance imbibed. Second is gradient of water potential between adsorbent and substance imbibed. Imbibition of water increases the volume of imbibant, due to which a pressure is created called imbibitional pressure. Next is wilting. On hot and dry days, plants lose more water by transpiration than they can absorb by roots. Turgor pressure is reduced and the cell becomes flaccid and the shoots and the leaves droop. This wilting is generally temporary and can be rectified by sufficient watering. In this image, we can see the wilting. During wilting, there is a large amount of loss of water. The turgor pressure is reduced and the cells become flaccid and the shoots and the leaves droop. Thank you so much. We will continue this chapter in the next video.